friend of mine was hiking at the Grand Canyon when the hike leader pulled her aside. He said, "I noticed your arms are not swinging while you walk. You should visit a doctor when you get home." Weeks later, she was diagnosed with Parkinson's disease. Parkinson's is a brain disease that's characterized by the progressive loss of motor control. It affects over 1 million people in the United States, and as 85% of patients have no family history of the disease, it can happen to anyone. In order to design treatments, we need to understand what specific cells in the brain are affected in the disease. Decades of Parkinson's research have focused on one particular type of cell, which is known to be destroyed by the disease. Unfortunately, by the time patients start to exhibit symptoms, many of these cells have already died, and they cannot be repaired or replaced. Current treatments targeting this aspect of the problem can provide temporary relief for some patients, but right now there is no long-lasting treatment or cure. I think we need to rethink the entire approach. What about the rest of the brain? I was very interested in whether there were other cell types affected later in the disease where we might still be able to catch the progression and intervene. Identifying these cell types, however, posed a particular challenge because the brain is extremely complex. It has many intermingled cell types that are each responsible for different functions. To navigate this, I developed technology that allows me to tag different types of cells with different colored fluorescent labels, as shown in this image. Then, within each cell type, I compared molecular measurements of the DNA, RNA, and proteins between healthy brain tissue and Parkinsonian brain tissue. My experiments implicated a second type of cell called PV cells that is particularly affected by Parkinson's disease. These cells are still alive in patients, but they have abnormal molecular behaviors that contribute to patient symptoms. Specifically, PV cells in Parkinson's disease experience changes to how they maintain oxygen and cellular energy. This presents an opportunity to develop an entirely new class of treatments that intervenes at an untapped node in the disease. It is likely that if we can target PV cells and correct their abnormalities, we can stop the symptoms of Parkinson's disease.